So in our previous chapter, we talked about what happens during non-disjunction when the mitotic mitosis process doesn't go quite as planned. In this chapter, we're going to talk about cancer, specifically the formation of cancer, which is also usually caused by problems with the cell cycle. So we'll start off with an overview of cancer. Cancer is simply uncontrolled cellular growth. Most cancers are a result from a cell accumulating mutations that causes a loss of control over the cell cycle. So basically that cell cycle, those stages of mitosis and meiosis, they just continue uncontrollably. So we have a single cell that keeps replicating and replicating and replicating and replicating, and usually it's replicating mutations along the way. There are a couple of key characteristics of cancer cells that allow us to differentiate cancerous cells from non-cancerous cells. First, cancer cells lack differentiation. Differentiation is the process by which a cell acquires specific structure and function. It's what allows a bone cell to be a bone cell, or a blood cell to be a blood cell, or a stomach cell to be a stomach cell. It's the differences in characteristics. So the cells that line your respiratory tract have cilia. The cells in your liver have increased amounts of smooth endoplasmic reticulum to help detoxify. So it's those specific characteristics of a cell that cancer cells tend to lack. Cancer cells are non-specialized. They don't contribute to the functioning of the body, and they tend to look distinctly abnormal. One of the things that's pretty abnormal about them is the nuclei. Cancer cells have abnormal nucleuses. A lot of times they're enlarged and contain an abnormal number of chromosomes. We'll see that they tend to have a lot of chromosomal mutations as well. Normal cells with damaged DNA undergo apoptosis, but cancer cells just simply don't. They don't really recognize the signs and the symptoms to undergo apoptosis, whether they are internal signals or external signals. They just simply ignore those signals and continue to divide. Tissues that divide frequently are much more likely to become cancerous. Cell division gives them an opportunity to obtain those genetic mutations, and then they pass on those genetic mutations for each round of cell division. So cancer cells have an unlimited potential to replicate. If they don't respond to apoptosis, they're going to just keep replicating and replicating and replicating. Cancer cells tend to be immortal, and they'll just keep on dividing. The ends of the chromosomes are repetitive DNA sequences called telomeres. So in normal, healthy cells, the ends of all of your chromosomes are capped with a sequence known as a telomere. In normal cells, those telomeres get shorter after each cell cycle. They do this with the presence of telomerase. Telomerase is just an enzyme that rebuilds the telomeres, and it prevents a cell from ever losing its potential to divide. So if we have that telomerase enzyme, it's going to keep attaching more DNA sequences to the end of that chromosome. With each round of with each round of DNA synthesis, those chromosomes get shorter and shorter and shorter. Once they get too short, the cell undergoes apoptosis, and it will not continue to divide because that DNA has just been copied too many times. You can kind of think of it as if you were to copy your entire textbook by hand, there's a good chance that even with a proofreader, you're going to make a mistake. It might not be a big mistake, it might not be a significant mistake, but you might make a single mistake if you're copying your entire textbook by hand. If you were to do that again, copy your textbook by hand again and again and again, and each time, instead of copying the original textbook, you're copying your copy, you're going to see an accumulation of errors. This is normal. It happens with all of our cells. The problem is not really a problem, but our bodies know this. We are aware that we're going to eventually make mistakes, and those mistakes are going to accumulate with time. And so what happens is after so many replications, the body shuts it down and says, all right, you have divided X many times, no more. You're not going to continue to keep dividing. We're not going to continue to keep replicating copies of copies of copies. And so we do this, we measure the amount of times that a chromosome has been replicated by the length of that telomere. The problem with telomerase is an enzyme that keeps adding DNA sequences to the end of the chromosome. So it's going to keep adding. With the addition of those DNA sequences, we lose our countdown clock that tells us how many times that DNA has already been replicated. And so those cells will just continue to divide into infinity. It's why we call those cells immortal. So telomerase, that's the enzyme that rebuilds those telomeres, and it prevents the cell from ever losing its potential to divide. And the gene that codes for telomerase is constantly turned up in cancer cells. Cancer cells also form tumors. Tumors are groups of cells. Normal cells can exhibit contact inhibition. Contact inhibition is when they come into contact with a neighbor, they stop dividing. Cancer cells don't do that. So with contact inhibition, the cell goes, it's a little bit crowded in here. I am being touched on all sides. I'm going to just not divide. It's plenty crowded enough in here. And cancer cells, on the other hand, are like, woohoo, it's a party. Look at how many of us are here. Let's just keep this party rolling. And they will keep dividing no matter how crowded they are or how surrounded they are. Cancer cells also disregard growth factors. Growth factors are chemical signals that come in two types. There's stimulatory and inhibitory growth factors. Cancer cells keep on dividing even when stimulatory growth factors are absent. So there's no factor that says, hey, you should grow now. There's just nothing. There's no signal. There's nothing telling them to grow. They're just doing it anyway. And they do not respond to inhibitory growth factors. So inhibitory growth factors start showing up. The body goes, hey, this is abnormal. There's a tumor over there. Let's shut it down. And it starts sending inhibitory growth factors. And the cancer cells just go, la, 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 I can't hear you. And they'll keep growing and dividing and growing and dividing anyway. So the next thing we're going to kind of talk about is the progression and the development of a tumor. So cancer cells gradually become abnormal. Cells don't just start off cancerous. It can take quite a bit for them to start to develop abnormal nuclei and then so cancer cells gradually become abnormal. It takes a little bit of time for cells to undergo the mutations and the mutations in the growth that are required to form a tumor cell. The process of developing cancer is known as carcinogenesis. It's a multi-stage process with 3 phases. There's initiation, so that would be like right over here. What happens is we have one cell mutates and it begins to divide repeatedly. So what you're seeing here is not this one cell spreading cancer to the cells around it. What's happening is this one cell is replicating in place. It's replicating in place and it's replicating and replicating and replicating. And so you can see that it starts to become very, very crowded and concentrated in that one particular area. So what happens is as that cell is replicating so quickly and so many times, it's going to eventually start to develop more mutations. So at this stage, it maybe has 2 mutations. And then the cell is going to continue to grow and divide. And each one of those cells that is growing and dividing has more mutations. And so those mutations keep mutating. It keeps growing. So that's first stage, that initiation. Promotion is when a tumor develops. Tumor cells are going to continue to divide and undergo more mutations in place. The problem is, even this isn't so much of an issue. What we'll have is a tumor that's growing in place. It's can become an issue if it starts pressing on vessels, if it starts pressing on blood vessels and blocking blood vessels, blocking nutrients from reaching other cells. It can become a problem if it is cutting off nutrients from other cells. But where it really becomes an issue is once the cell starts to become invasive. Once the cell gains the ability to invade surrounding tissues, it becomes very dangerous because it can reach into the bloodstream. If it can access a blood vessel or access a lymphatic vessel, it can then travel to various parts of the body and start developing in other areas. So just because cancer is a very personal issue, most individuals know someone who has experienced cancer. I will share a lot of personal stories through this time just because that's what I have. So my mom was diagnosed with breast cancer a few years ago. And what she noticed, what was initially noticed, was large tumors that were developing in breast tissue. The very first thing that they did was check the margins of the tissue. They were looking to see that the tumor itself was nice and rounded and not jagged. When you have a jagged tissue or a jagged tumor, it's an indicative of invasion into other tissues instead of a tumor simply growing in place. Fortunately, the margins of her tumors looked really good. So although there were multiple tumors, they did not look like they were invading surrounding tissue. They had not quite metastasized. And so they weren't, there was less concern that they had invaded tissue. Even though they were certain that they had not invaded tissue, the next thing they looked at was the location of those tumors to lymphatic vessels. As we've already talked about, lymphatic vessels can transport throughout the entire body. And so there is a major concern if a tumor is growing near or around either a lymphatic vessel or a circulatory vessel, because that tumor can then spread into other areas of the body. Since her tumor margins looked good, there was no invasive tissue. There was no invasive tumor tissue. They were able to just excise the tumor. They cut the tumors out. And then they were also convinced that the tumors were sufficiently far away from any vessels, that they were not concerned of any sort of metastasis of the tumors in other parts of the body. So because of that, she did not need to undergo any sort of chemotherapy or radiation. She just had to undergo many, many doctor visits to double check, make sure that the tumors had not grown. She did opt for a double mastectomy to ensure that there was absolutely no chance of any remaining cancer cells to be found in that breast tissue. So a mastectomy, a double mastectomy, is when they simply remove all breast tissue, which is just to further ensure that there is no tumor cells left behind. So we're actually going to pause right there, and we will pick up with angiogenesis and metastasis in our next video.